Our university is one of the biggest and oldest university and hospital. Actually, it was built by King Gi-Jeong in the Joseon dynasty. I think that old history and basic foundation is really important for our students and the faculties.
One of the strongest points in our team is that we have a very different background, and faculties in here, including the physics engineering, as well as radiopharmacy and the radiochemistry.
Our department is affiliated to the clinic, so actually our lab is consist with the clinicians and the PhDs. And we have 3 PhD PIs. One is, for me, is a biology teams.
In our campus, we can work together because we have our university and Cancer Research Institute and Clinical Trial Center and Animal Study Facility, and also for the cell line bank are all together in this campus.
Some of our project actually is in the clinical trial. We developed some kind of imaging method to tracking the mesenchymal stem cells. Actually, the stem cells has some kind of therapeutic genes. The cytosine deaminase, it can convert 5-FC prodrug to the 5-FU drug. And actually the mesenchymal stem cells has a characteristic to chasing the tumors. So we use the glioma, and mesenchymal stem cell which express the therapeutic gene can chase to the glioma, and then when we treat the prodrug and it convert to the drug, so it's very specific targeting to the gliomas. Stem cell trafficking is really important because the stem cell is really useful for the treatment of certain disease. So the most important thing is that kind of a therapeutic cell can go to the target.
Using the molecular imaging, we can validate and forecast how the biomarker targeted drug is working on individually in each regions.
For development of new radiopharmaceuticals, discussion with the nuclear medicine physician is very important.
The main goal is that to develop a new imaging biomarker as a multiparametric imaging systems.
When you treat the patient using the targeted agent, sometimes it took about 1 month or 3 months after treating the patient to get the result of the response. But when we're using the molecular imaging or PET-CT, you can know only 1 week or 4 weeks after treatment starts. So earlier response is quite beneficial or informative for the clinician to determine the future treatment.
In Korea, we opened the first PET center in 1994, and PET became a major clinical application of molecular imaging in this country. In statistics, I heard that last year we have about 250,000 clinical PET study.
For diagnostic radiopharmaceuticals, we develop PET radiopharmaceuticals such as F18, C11 or gallium-68 labeled agents, and we also develop technetium labeled radiopharmaceuticals for diagnosis. For therapy, we worked with rhenium-188 more than 10 years ago, but recently we are more concentrating on lutetium-177, and we also interested in yttrium-90. These are beta emitters, and we are also interested in alpha emitters such as actinium-225 and astatine-211. But those radionuclides are difficult to obtain. So I think tin-117M, which is a convergent electron emitter, may be very important for therapy. So now we have special interest in tin-117M labeled radiopharmaceuticals for therapy.
We have developed and commercialized the preclinical small animal PET/MR scanner, and currently that we are focusing on developing the clinical version of the kind of simultaneous PET/MR machine. Now that we are developing the human brain PET insert for 7 Tesla PET/MRI. We apply new machine learning algorithms for improving the PET image quality and the MR image quality. We use those kind of algorithms for improving the quantitative accuracy of our molecular imaging systems.
We want to evaluate that metabolic activity or the physiologic activity from the PET system, and also with physical chemical characterizations from the MR system. So in that kind of point of view, so some simultaneous acquisition is very important, especially in the animal or the basic experiments.
I would say that the PET/MR has several advantages that include the better soft tissue contrast of MRI and smaller radiation dose than the PET/CT, and different from the PET/CT. Actually, we can simultaneously acquire PET and MR images together.
The WMIC is the biggest congress in the world for the molecular imaging. Also, they touch on very different areas, including different technology, cutting-edge technology, such as MRI and photoacoustics and radionuclide and optical, as well as that they also touching very different, focusing at the application areas such as infection, inflammation, oncology, and urology, cardiology.
Seeing is believing. We can explain lots of things in the mechanism or indirect method, but the best way is to show what's going on. And the good thing is that the molecular imaging societies, they can give me a lot of inspirations. How can I visualize this biological response? And we can exchange a lot of information, and we can exchange the vectors or transgenic mouses. I got a lot of help from the many members of the Molecular Imaging Society. That's why I attend the WMIC.
여기가 우리 연구실입니다.
여기가 우리 랩입니다.
여기가 우리 실험실입니다.
여기가 우리 실험실입니다.